Banan and Alnemri, and this work was done in collaboration with Arwa Basbrain from King Abdulaziz University in Jeddah, Saudi Arabia. Today, I will be presenting our research titled Automated Morphological Analysis of Neurons in Fluorescence Microscopy using YOLOv8. This presentation will walk you through the motivation behind our study, the methods we used, the results we achieved, and finally, how this work contributes to the future of biomedical imaging. As shown here, I'll begin by clarifying some key terminologies used throughout the paper. Then I'll introduce the background and related research in morphological cell analysis. After that, I'll explain the YOLO version 8 model, the dataset, and our methodology in detail. We will go through the measurement equations, visual results, evaluation matrices, discussion, and end with our conclusions and future directions. Before diving into the technical sections, it's important that we share a common understanding of some of the key terms used in this study. Fluorescence microscope is a technique that uses fluorescence dyes to visualize and capture detailed images of cellular structures. A neuron is a nerve cell responsible for transmitting electrical signals in the brain and nervous system. Segmentation refers to dividing an image into regions to identify each cell separately. YOLO version 8 stands for You Only Learn Once version 8, which is a deep learning model for real-time object detection and segmentation. Instance segmentation means that the model not only detects an object, but also outlines it individually. Morphological features are the physical characteristic of a cell as its length, width, area, and intensity. Ground truth represents the manually annotated verified data we use to check the model's accuracy. And finally, intensity refers to the brightness level of each pixel on a 0 to 255 scale. These terms will appear repeatedly, so keeping them in mind will help follow the rest of the talk. The foundation of this research lies in morphological analysis, or simply put, measuring and understanding the structure of cells. Quantitative analysis of cellular morphology is essential in biomedical research, diagnosis, and therapy development too. So by measuring cell size, shape, and internal components, researchers can detect structural changes linked to disease such as cancer or infectious diseases. However, traditional approaches often rely on semi-automated image processing tools that still need manual correction and expert supervision. These methods are slow, prone to human variation, and not scalable for large datasets. But in contrast, deep learning techniques now allow fully automated and high-throughput cellular analysis. Among these, the YOLO family of models has become very popular because it combines speed and precision, so making it ideal for real-time medical image applications. To expand a bit more on the context, cell morphology provides critical insights into biological process. Accurate measurement of features like cell size, area, and intensity can indicate cell health, differentiation, or disease progression. As we mentioned, traditional analysis methods such as manual annotation or semi-automated segmentation are labor-intensive and inconsistent across observers. So there is a strong need for automated systems that can perform these analyses quickly and accurately. Deep learning, particularly convolutional neural network, has shown remarkable performance in this area. That's why our research leveraged the YOLO version 8 model, which is the latest and most advanced version, to not only detect and segment neurons, but also quantify their morphological properties automatically. The need for accurate, scalable, and repeatable quantification of cellular features has driven the growth of the field of automated cell analysis research. Cellular structures have historically been segmented and measured using conventional image processing methods like thresholding, watershed segmentation, and morphological filtering. However, these techniques frequently have difficulties with complicated biological images, which result in inconsistent segmentation outcomes. Deep learning approaches have significantly improved the accuracy of cell detection and segmentation, as in biomedical image segmentation tasks, CNN-based architectures like Deep Lab version 3 Plus and UNIT have shown superior performance. These models use encoder-decoder structures to extract spatial features and generate precise segmentations. Furthermore, for cell localization and counting tasks, object detection frameworks such as SSD and faster RCNN have been used. Now let's focus specifically on YOLO version 8, which is the backbone of our study. YOLO version 8 is highly optimized for high-resolution biological images like fluorescence microscope. It combines improvement in both object detection and instant segmentation at the same time, meaning it can accurately locate each neuron and draw its precise outline. Previous research has already used YOLO models to detect cells in fluorescence and histopathological images, proving the model's potential in biomedical imaging too. However, there is still a lack of comprehensive evaluations, especially those that assess how accurately YOLO can reproduce morphological measurements such as cell length, width, area, and intensity. So our research addresses this gap by providing one of the first detailed evaluations of YOLO version 8 measurement accuracy using a high-quality annotated neuron dataset. Our methodology consists of two main components, the dataset we used and the model training and measurement process. So let's start with the dataset. The dataset contains more than 17,000 cells distributed across 90 high-resolution 2D fluorescence microscope images. These images capture neural-like cellular structures derived from stem cells. The dataset was originally derived from an earlier study for cell segmentation and phenotype analysis. We created the dataset in an earlier work, and we reused it here to evaluate YOLO version 8 capability of replicate accurate cell measurements. The main objective of our paper is to determine how effectively YOLO 8 can reproduce these morphological measurements for biomedical image analysis. So before the training, we performed several pre-processing steps. First of all, images were converted to grayscale, simplifying intensity analysis. Pixel values were normalized to 0 to 255 range. All images were resized to maintain a consistent scale. For the training, we trained an instant segmentation YOLO version 8 model using the ultra-text implementation with default hyperparameters. Ground truth annotations from V7 Darwin were exported in JSON format and then converted to binary mask using OpenCV and NumPy libraries. This created a consistent environment for both training and evaluation. The morphological measurement for each segmented object in both ground truth and predictions were captured as you can see the equations on the screen. Cell length and width, the height of each object's bounding box was used to estimate its length and width. We obtained X, Y, width, height for each mask using OpenCV's function. Then we noted the height as the cell length and the width as the cell width. This method guaranteed consistent length and width estimation across irregularly shaped cells. For the cell area, we used NumPy's function. The area was calculated by counting the number of non-zero pixels in each binary mask. Using this formula, where this one is in the indicator function that equals 1 when a pixel belongs to the mask. So the pixel count reflects the number of pixels belonging to the cell instance within the mask. This figure illustrates how we extracted morphological parameters. Each segmented neural light cell is enclosed in a bounding box. The arrows show length along the major axis and width along with the shorter axis. The area corresponds to the total number of pixels inside the mask. So this visualization helps clarify how morphological parameters were measured objectively from each segmented cell. Now for the intensity, the binary mask was used as an index on the original grayscale image to extract intensity values. We gathered all pixel intensity values inside the mask region using NumPy. Three NumPy's functions, actually, as you see, were used to calculate the mean, the minimum, and the maximum from the pixel array, where each I of j is the grayscale intensity at pixel I and j, and the mask is used to select only relevant pixels. To ensure consistency between training and evaluation, we took the following steps. All images were resized, so every measurement used the same resolution. Also, for the intensity values, they were recorded on 0 to 255 grayscale. Length and width were measured in pixels, but the area in square pixels. Measurements from all detected instances, ground truth and predictions were combined for each image. Ground truth masks from V7 Darwin were exported and converted into binary masks. The same measurement functions were applied to both YOLO version 8 predictions and for the ground truth at the same time. These steps guaranteed that both pipelines were identical, eliminating methodological bias and ensuring a fair comparison. Throughout the dataset, two main evaluation criteria were used to evaluate the YOLO version 8 model's performance. The first one is the segmentation performance, and the second one is the measurement accuracy. When combined these two, it enables us to assess the model's ability to accurately reproduce biologically significant measurements like area, length, and intensity, in addition to its ability to recognize neuron instances. To evaluate segmentation quality, we use the following standard matrices: precision to measure the proportion of correctly predicted cells out of all predictions made, recall to measure the proportion of correctly predicted cells out of all actual cells. The F1 score represents a balance between precision and recall. Intersection over union to evaluate how well the predicted mask overlap with the ground truth mask. The model showed an overall accuracy of 97 percentage. As you see at table 1, the segmentation matrices results. Now, for the measurement accuracy. To assess measurement accuracy, we compared each predicted morphological value with its corresponding ground truth value. So this process was repeated for every matched cell in the dataset. For each matrix length, width, area, or the intensity values, we calculated the percentage accuracy using this formula. We then averaged all these accuracy scores to determine the overall measurement accuracy. As you can see here, these measurements demonstrate YOLO version 8 ability not only to segment cells accurately, but also to provide quantitative data that are biologically meaningful. While some matrices, such as minimum intensity and area, were less accurate due to noise and low signal regions, other like the mean and maximum intensity were nearly perfect. You can see here at the figure provides a visual comparison between the original Fluorescence microscope image, the manually annotated ground truth mask, and the YOLO predicted segmentation. In this example, the ground truth contained 136 cells, while YOLO detected 189 cells. Although a few over the detections occurred, the model effectively captured most neuron boundaries, showing its robustness in complex images. So let's discuss these results. Overall, YOLO version 8 performed robustly in both segmentation and measurement tasks. It achieved a high accuracy for mean and maximum intensity, showing excellent preservation for brightness information. Although area and minimum intensity were slightly less accurate, likely because of weak signals and overlapping cells. One major strength is that our pipeline integrates segmentation and measurement into a single automated framework. However, there are still some limitations, such as handling illuminated and or overlapping cells and the relatively small data size. We proposed an automated framework for neuron segmentation and morphological analysis using YOLO version 8. Also, the model achieved 97 segmentation accuracy and 75 overall measurement accuracy. This approach enables large-scale and objective analysis with minimal manual effort. Future directions include expanding the dataset, refining segmentation for complex neuron structures, and integrating biological validation matrices. In summary, our work bridged the gap between deep learning and practical biomedical imaging by demonstrating how YOLO version 8 can perform both segmentation and quantitative measurement efficiently. This research was inspired by an earlier study by Basbrain at 2022. That work focused on converting stem cells from developing wisdom teeth into functional neurons. Our dataset originates from those experiments, providing high-quality fluorescence images for training and evaluation. These are some of the main references that supported our research, including foundational works on YOLO unit and Deep Lab version 3, as well as the biological source study. Thank you very much for your attention.